Professor A.K. Sharma sir, Professor and Head Department of Biotechnology. Without his blessings, it was not possible to organize this event. And indeed, matter of pleasure for us to hear him. He is a very learned personality in cancer research, epigenetics, biomarkers, given by his credentials by publishing a very good impact journals and books, many more books in his credit. So over to Professor Sharma sir. Sir,
Thank
please,
you.
sir.
Thank you, Dr. Sheel, for such a nice, elaborative introduction. Thank you so much. And I would be going to my talk by sharing the screen. Siddharth, can you allow me for sharing the screen?
Yes, sir.
Okay.
What's a screen share?
Okay. Is it visible, sir?
Yes, yes, sir. Yes, sir.
Okay. Okay. So, thank you, Dr. Sushil, and the previous, our chief guest, Dr. Malhotra, for being so elaborative about his work throughout his career. And I thoroughly enjoyed that talk as well. And the title of my talk for today is the molecular diagnostics in the breast cancer area. This was the work one of my research scholar he performed. Before going, I'd like to revisit the campus. It's located at Mullana, Ambala, and it's a beautiful, serene campus spread around 200 acres of land. And this is another view of the campus. And before moving on to the talk, let's devote these few quote by Dr. A.J. Abdul Kalam, that if you want your country to be corruption free and a nation of beautiful mind, there are three key societal members who make a difference. That is father, mother, and the teacher. So these three are the stakeholders, the pillars of our moral ethics and moral values. And two things are infinite in this world, the universe and the human stupidity. And I'm not sure about the universe. These were the words of the Albert Einstein. And there's a very interesting lineage in the history. If you see the series of philosophers produced as a result of this mentor and mentee relationship. So we should attribute these etiquettes towards establishing this kind of mentor and mentee relationship. And the great Mahatma Gandhi, the father of nation, he said these 7 deadly sins we should never commit, and science without humanity is one of them. So we should never do any science which does not have applications towards humanity. So these are the thrust areas. Our department here in Mullana is pursuing into diverse areas in the industrial, environmental, medical microbiology, biotechnology, and nanomedicine, cancer biology. And my focus of this talk is on breast cancer therapeutics through biomarkers. And we were focusing on diagnostic markers, prognostic markers, or the pharmacodiagnostic markers. And when it comes to breast cancer, these are the words of Debbie Wasserman Schultz. Every woman needs to know the facts, and the fact is that when it comes to breast cancer, every woman is at risk, and there can be life after breast cancer. The prerequisite is the early detection through these markers. So even this is the Angelina Jolie, who had this 87% risk of breast cancer and 50% risk of the ovarian cancer, but it was detected at the early stage through these markers. So that really intensifies the significance of these markers. So let's understand what are the risk factors of these this cancer. There are multiple factors: age, to the sex, to the radiation, chemotherapy, genetics, and many hormonal metabolic factors and environmental lifestyle factors. And medical complications also sometimes result into cancer. And there could be direct factors and indirect factors also, contributing factors, and some vulnerability factors also. So in direct factors, you see radiations, inherited mutations, and in vulnerability factors like hormonal imbalance, early menses or late menopause, and no pregnancy or lactation. And then there are some lifestyle and other contributory factors as well. So coming to the basic definition of the markers, what are markers basically? Many cancers, we have seen that they are associated with abnormal production of some molecules, which can be measured in the plasma. So you can easily distinguish them, and based on those, you can diagnose a particular form of carcinogenesis. So we targeted to these breast cancer biomarkers and looked into three particular marker genes, that is ERBB2. Then there is a breast cancer associated gene BRCA1 and TP53. So all these three are located on human chromosome 17. And why we targeted those only? Because in the previous studies, we found that they seem to be upregulated in cancer patients, breast cancer patients. So what we did was that since this scholar was from and he brought some samples from there because the incidence of breast cancer was pretty high at his place. So we wanted to compare that population with the population here in northern India. We took some samples from the PGI Chandigarh and some from the medical hospital at Mullana itself. And though the sample size was not that high, we could approach only up to 50 patients from this particular region and 50 patients from the Baghdad area. So, and wanted to see whether the pattern, the regulation, the level of expression of these markers is similar or dissimilar, or it is different in both these populations. So, and then followed by not only the level of expression, gene expression, but also we looked into the gene sequencing part so as to see whether the pattern of the mutations, mutagenesis, is similar or dissimilar in these two kinds of populations, Middle Eastern and the North Indian population. And then we try to correlate to establish some relation amongst these markers with the breast cancer pathogenesis. So the very first thing we looked at the age factor, and what we found was that in both Indian and the Iraqi population, the 40 to 49 years of age, when the female reaches toward the menopausal age, that was the most vulnerable population for prone to breast cancer. So 40 to 49 years of age on an average in both the populations were quite prone to breast cancer. And these three overall, they seem to be tissue specific markers because in blood, we did not see their upregulation, or their levels were significantly lower in comparison to the tissue. So more or less likely they seem to be tissue specific markers than than the blood. So when we compare these three levels, like we took the BRCA1 gene here and looked at both the populations, though the error bars are pretty high, but in both the populations, Indian and the central Iraqi Baghdad population, we found that there was not much significant difference in terms of fold change in expression of BRCA1. And though if you see the malignant cases, the level of mRNA expression was significantly higher in comparison to the benign or the control, so healthy control. So we found that in both the populations, these three markers, TP53 to BRCA1 to ERBB2, the level of expression was significantly higher in malignant tissue samples in comparison to the benign or the healthy controls. So you see this comparison in all these cases, and this is the comparison in blood samples where we don't see any significant difference in malignant and the control or benign samples. So that shows that these three markers were tissue specific, and that's why their level malignant tissue samples was significantly higher in comparison to benign and the control. So ERBB2, when we compare this epidermal growth factor receptor gene, which is a viral oncogene, the name is derived from erythroblastic leukemia, viral oncogene. So here also Indian and Iraqi population, the mRNA expression was pretty much similar, you can say, because there was no significant difference in Indian and Iraqi population. So up till here, what we can say is that the three markers which we have chosen, they were tissue-specific markers, and the malignant in the malignant tissue samples, the level of these markers was significantly higher in comparison to the control and the benign samples. While the two populations which we took, we did not see much differences in terms of expression. And when we looked at their sequence profile, so we had to run the DNA exons on the gel and looked at their sizes and then sequenced them. So when we amplified this TP53 exon, we took one exon, hotspot region, exon 5 actually. Why we took exon 5 here in TP53? Because this was previously reported to have significant impact when there is breast cancer. So we were expecting a size of 520 base pairs in Indian and population on the left side and Iraqi population on the right side. So almost at the same level, 520 base pairs, we got this DNA amplification also. And the ERBB2, here also we were expecting a size of 490 base pairs, and Indian and both the populations, we got the amplification at the same spot. And the BRCA amplification, here we targeted the exon 16. So here also we were expecting a size of 370 base pairs, and which you can see this amplified band. And then we looked into the sequencing part. We sequenced this and looked at various changes, mutations, and we found mutations in both the populations. So if you see this compiled data, we found some mutations in Indian cases and the other population also. But this data still needs, we need a larger cohort of studies, larger sample size. to establish this correlation because there was lots of polymorphism and the mutations were observed in both the populations. Like these are some of the changes we saw in ERBB2 exon 16. There was some mutation at this spot, and similarly in TP53 exon 5, and then the BRCA1 also. So overall, what we see is that the tissue-specific expression is there of all these three markers in both the populations, and the levels are significantly higher in malignant tissue samples in comparison to the healthy controls and the benign samples. And hotspot regions, exon 11 of this BRCA1 and exon 5 of TP53, exon 16 of ERBB2, they were more prone to mutations, changes in both the populations. And there is a differential gene expression of BRCA1, ERBB2, and TP53 at mRNA level. So we propose that these markers are good diagnostic markers, especially having a significant prognostic value in breast cancer patients and breast cancer therapy. So they can be strong candidate for strategizing the therapeutic intervention in breast cancer. So the lifestyle, it is related to lifestyle. So what our recommendation is that people should maintain a healthy lifestyle, and they should be more into the intake of antioxidants through fruits, oils, milk, and meat and bean, and daily physical activity is must. And also at work, many of our population, they are exposed to carcinogens. So we should try to maximally avoid exposure to carcinogens at our workplaces also. And these were some of the publications. More recently, we tried to look into the microbiota, also human microbiome, and what are the dietary factors and environmental factors, how they contribute and how they maintain the microbial homeostasis and the interactions thereof in the microbiota and influencing the health, overall health and carcinogenesis also. So these articles we got published into good impact journals, and this is another paper we published recently, and establishing a tripartite linkage between dietary factors, host mucosa, and microbiome, which is a next generation now therapy in terms of personalized medicines. So these are my research scholars. I haven't put one more, Dr. Varuchi Sharma. I could not put his picture on here, but these are the other research scholars who passed out under my guidance. And some of my recent publications in the cancer area. And some of the books we have published recently. And I was invited for some expert talks at various places. Once again, I profoundly thank you all for your kind attention, and I hope you enjoyed the talk, and it would be open to questions also if you have. Yeah. Thank you again. Thank you for your patient listening. Thank you.
Thank you, sir.
Thank you. Thank you, Dr. Raj. Yeah.
Thank you.
Any questions? I would be happy to answer.
If you have any question, please may ask.
Excuse me.
Most welcome.
I am Professor V.K. Bhattu from Himachal Pradesh University, Shimla. I want to congratulate Professor Anil Sharma for such a brilliant talk on breast cancer. Earlier
Welcome,
also,
sir. Thank you.
I wanted to congratulate Professor Sandeep Malhotra. My link was broken that time, for getting the fellowship from this prestigious university. Both were very brilliant talks, and we thank both the speakers for such nice interaction in the morning. Thank you so much.
Thank you, Dr. Vinod, for this much appreciation. We profoundly thank you, sir. Thank you.
So, thank you, sir, for such outstanding, informative, illuminative talk. And
thank you, Dr. Sushil. Thank you so
now
much.
it's a moment to once in our life, I suppose, I'm very young generation for you because I hear very little time. So I wish to felicitate Professor Sharma sir by Fellowship Award of International Association of Zoologists. So I wish
Thank
to
you, Dr.
request,
Sushil. Thanks a lot for this honor.
I wish to request Professor Muttu sir and Professor Malhotra sir kindly felicitate him by this kind of fellowship award. Sir, kindly share your screen, Sharma sir, please.
Okay. Should I turn it off?
Yes sir, yes, unshare kar dijiye sir. Stop sharing.
Okay. All right.
So on behalf of organizing team, by Department of Biotechnology, and on behalf of IAZ, we wish to felicitate Professor Sharma sir.
Okay, congratulations, sir.
Thank you, Dr. Raj. Thank you so much. Thanks a lot. Yeah, thank you.
Very nice talk and very informative talk.
Thank you, sir. Thank you.
Congratulations, sir. Congratulations for this talk.
Thank you, sir. Thank you. Thank you so much. Thank you, Dr. Anil.
By these words, we wish to congratulate Professor Sharma sir.
Excuse me, please.
Yeah, we have one more
hello.
speaker.
Yeah,
Ji, ma'am.
I'm Dr. Neela Mattu. I want to talk to Dr. Anil Sharma, please.
Yeah, please.
Hello,
please.
ma'am.
Yes.
Yeah. Very good morning, ma'am.
Congratulations. Very good morning. Warm greetings to you. It was
Thank you,
really
ma'am. Thank you so much.
such a productive talk, which is need of the hour. Actually, such awareness is needed very early for the
Right,
girls,
ma'am. Right.
especially at very young stage. Because in our country, you know, so many things are being hidden. We just keep
Right,
hiding
ma'am.
such
Right.
things. Yeah.
Yeah.
And I would like to share here that one of our friends, even whose son is doctor, she had some bleeding from her breast, and she didn't tell it to anybody of the house. And you know, after 5 years, she just passed away because it was so much cancer. It was spread badly. So it is something that which is not to be hidden. And if there is any problem, it should be at once. It should be checked out and to good doctor and all. And because it is just the risk of the life.
Yes, ma'am. There are many.
So thank you so much for such a wonderful lecture.
Yeah,
Thank you so much.
There are.
Okay, congratulations.
Thank you, ma'am. What I see is in our system, we have many social enigmas, social hindrances, because of which we try to hide. If we see the example of Angelina Jolie, who had like 87% risk of breast cancer and the ovarian cancer, but she did not hide it and went to the doctor at the early stage and diagnosed it successfully through the biomarkers, and now she's all right. Yeah. So that's what is challenge for our country and the other Muslim countries also, like Middle East and the many Asian Muslim countries also. So many social and religious enigmas are there, and what the recommendation is that when it comes to cancer, we have to target it at the early stage. Only then we have some solution. Otherwise, we get to know all about this when we are at a stage where it is difficult to stop the proliferation.
Yeah,
So,
very true, very true.
so overall recommendation is that let's
Yeah.
go early. Whenever you see any of the symptoms, and after the age of 40, I think every woman should get her mammogram done in a year, and also the puberty stage, like early stage, the girls, when there is this upregulation of hormones, they should be very careful. If they see any symptom, they should directly report to the physician. Yeah, thank you, ma'am. Thank you so
Thank you,
much
sir.
for
Thank
the
you so much. Thank you so much.
Thank you. So over to Siddharth for the next proceedings. Yeah, go ahead.
Yes, sir. Thank you so much. Now
Thank
we
you,
have
doctor.
the next speaker from DRDO, Professor Mansu Ojha, sir, senior scientist. He will be with us after half an hour. I suppose we will start the next session at 12 o'clock. Okay, so this is a short break for us, and we will get together once again at 12 o'clock with Professor Ojha, sir, from Senior Scientist DRDO.
Okay, okay, sir.
Okay, sir. Sir, Sharma, sir, may I?
Yes, Dr. Shil.
Yes.
So,
Sir,
yes,
it's a break
yeah.
for half an hour,
So
sir.
we are going to for a break. Yeah. Okay. Go ahead.
Yeah, yes, sir.
Yeah.
Yes, sir. Okay.
Thank you.
Thank you, sir. Thank you so much.
Thank you.
Thank you all kind participants, faculty members, our colleague, senior respected colleagues, Professor Mattu sir, and all other invited delegates and guests. Thank you for your kind presence here at the early time, because it's the second Saturday. Most of the colleagues are on leave. I'm taking their quite more busy schedule and engaging them. So thank you for your kind presence here. We will meet once again at 12 o'clock.
Okay, sir.
Thank you. Thank you.
Thank you.
Thank you.